Assalamu alaikum. I hope you guys are all doing great. I'm gonna talk today about transcription and translation, and we're gonna go briefly from the paragraphs of the book, and then I'm gonna summarize it with these pictures. Okay, so to start with transcription, which is this we have here a spoken message that's converted to a written copy. It transfers the information from a DNA-based sequence to an mRNA-based sequence. Okay? Here they are saying that the form is different, but it has the same information. And now we're gonna talk about what do they mean by this. How is the form is different and how does this happen? So once the mRNA molecule is made, it attaches and leaves the nucleus via a nuclear pore and heads for the protein synthesis machinery, which is the ribosome. So first, the mRNA is made in the nucleus and it cannot leave until it's mature mRNA. And how does it leave? It leaves through the nuclear pores to the cytoplasm to start protein synthesis. Okay, so we can say that mRNA happens in the nucleus, and which is transcription, and protein synthesis happens in the cytoplasm, which is translation. And the ribosome, which we all know, this whole process cannot happen without the ribosome. It has a small subunit and a large subunit. So transcription cannot begin until what? There's a few steps that should happen before transcription starts. Gene activating chemicals called transcription factors stimulate histones at the gene transcription site to loosen. So we all know what are histones. They are like small histones that keep the DNA compacted and it should be loosened. This happens by the transcription factor. It stimulates it. Tamam. Then the transcription factor binds to the promoter. This promoter actually tells it's here to tell them that come to me, I'm here, yalla, let's start. Okay, so a special DNA sequence that contains the start point where we should start transcription. Here is it, like here it's the promoter region. It tells the RNA polymerase, come, I'm here, let's start transcripting. Tamam. It also specifies which DNA strand is going to serve as the template. As it tells them like, come to me, I'm here, it specifies to them like, I'm the template strand, start with me, as an example. So the uncoiled DNA strand not used as a template is called the coding strand because it has the same coded sequence as the mRNA to be built. Okay, I'm gonna talk about this Okay, so we have here 2 DNA strands. These are okay, here it's uncoiled and it started. Okay, هذا ال RNA polymerase إجا. هاي ال template strand وهاي ال non template. Okay, template non template وال mRNA strand. فال mRNA strand بتاخد ال informations من ال non template one. ليش عم بيحكوا لنا ال template هي coding strand؟ لأنه it has the same information as the mRNA strand بس in a different way. Why? Because in DNA we has هدول عنا بالsequence اللي عناهن هن الأدينين سيتوزين جوانوسين وثيامين. بس بالmRNA عنا أدينين سيتوزين جوانوزين ويوراسيل instead of thymine. This is the difference between DNA and RNA. تمام. Okay, so if they ask you what's the difference, then that mRNA has a uracil and DNA has a thymine, and they may give it to you as an example of like if they ask you give me the مثلا give me the coding strands sequence أو مثلا عطوكن UATC ما في سي okay wait UACG وبدهم يطلبوا العكس. تمام. هلا هون نعطيكم السيكونس هاد كنت تعكسوه هيك as AUGC بس لو عاطينكن اياه بالدي ان اي بدكن تحولوا للام ار ان اي، لازم ما تنسوا انكم انتوا بدكن تحطوا بالام ار ان اي 1 a U instead of a T. تمام، لانه ممكن يحطوا لكم بالاوبشنز هي السيكونس فيه T which is بالام ار ان اي مو موجود، بدكم تنتبهوا على هاد كمان. هلا هلا ال RNA polymerase ال RNA polymerase هي اللي بتجي وبتبدأ البروسيس itself. تمام، it initiates transcription. هي ال RNA polymerase it comes بتمسك ال two strands و it starts the process. تمام. Okay, so we have three phases: initiation, elongation and termination. The processing itself قبل ما نبدأ في عنا editing و further processing, cleaning up the raw mRNA transcript because the DNA is transcribed sequentially. The mRNA initially made called the pre-mRNA. Okay, هون في شغلة عم بيحكوا لنا they are talking about the mRNA itself, that before it goes to translation it's a pre-mRNA. Why is it called pre-mRNA? Because it's not ready yet. So the pre-mRNA, if we're gonna talk about the pre-mRNA, this is the pre and this is the mRNA. بشو بيختلفوا؟ They differ in that the pre-mRNA has exons and introns. تمام، but the mRNA has only exons. okay, شو هي الexons والintrons؟ exons are the coding شو اسمه، they have the coding informations بس الintrons non-coding، فمشان هيك they should be removed before it goes out of the nucleus to translation, which is in the cytoplasm. ف splicing process happens where introns are spliced out and exons are connected together to produce a mature mRNA. This one is a immature mRNA and this one is a mature mRNA with all the coding infos. تمام. With what does it happen? It happens with a spliceosome. I'm gonna mention it just in a bit. So the introns must be removed before it gets out of the nucleus to the cytoplasm. Here, the spliceosome. This spliceosome, it snips out the introns and splices together the remaining exon-coded sections. So the coded sections are the exons and the non-coded sections are the introns. That's why we remove them to produce a mature mRNA. If they ask you, they may ask you like, what removes the introns or what splices the introns? You should know that spliceosomes is responsible for all of this, producing a functional mRNA. Now our messenger is ready to exit the nucleus and to enter the cytosol, participate in the translation. So we all got that mRNA should be mature before it leaves the nucleus. And how does this happen? It happens by the spliceosome. It cuts the introns and connects exons together and produces a mature mRNA. This is all about transcription. Let's summarize it in these pictures. So first, we all know that a gene is a segment containing nucleotides that starts with the promoter and ends with the terminator, and transcription occurs in the nucleus and translation occurs in the cytoplasm. Now for initiation, the promoter region functions as a recognition site for RNA polymerase. تمام. So RNA polymerase comes, the promoter functions as a recognition site in the start point. Then it binds to it and the double DNA helix unwinds. In the elongation here, the RNA polymerase slides along the DNA, and once the RNA polymerase reaches the terminator region, the mRNA is complete. Okay, here as you guys can see, it slides, and here we have the coding strand, we talked about it, and the template one. Here's the mRNA, it slides out with the RNA polymerase. So the mRNA comes up with exons containing coding proteins and introns, and we said that introns should be cut out and we should connect the exons together. Intron splicing happens using the spliceosome to produce a mature mRNA that leaves the nucleus through a nuclear pore and enter the cytoplasm to begin translation. That's it. Here it comes. When it reaches the terminator region, the RNA polymerase and the completed mRNA detaches and it's degraded. What else? Yeah, you can read what's written here, but just briefly. Now translation, which is step of protein synthesis. The language of nucleic acid is translated into a language of proteins, amino acid sequence. The genetic code, the base sequence of a gene, is translated into an amino acid sequence called the genetic code. For each triplet or 3 base sequence on DNA, the corresponding 3 base sequence on mRNA is called a codon. Okay, so this is the codon. These are the codons. وفي من هون anticodons كمان. Since here are 4 kinds of RNA nucleotides, there are 64 possible codons. هون عم يحكي لنا هون كمان that each amino acid has multiple codons لأنه we have only 20 amino acids, and to produce as much codons as we can, so we use multiple codons on one amino acid. It's written here, I guess. Yeah, some are specified by more than one codon. It helps protect against problems due to transcription errors. Tamam. We have also the most important codons that we all should know and memorize, which is the stop signs and the start signs. Stop signs that call for termination of a polypeptide synthesis. The stop signs, they all start with, there are 3 only. They all start with U and they have A and G. But there is only one that is UAA, and the other 2 you can just use it as UAA and then UGA and switch between them, UAG. So UAA, UGA and UAG. And for the for the start sign, it's AUG. And how did I memorize it? As an August. So August is the start sign, which is AUG. Tamam? These are the only ones that you should know, and I don't think that you should know this, but AUG, which is the start sign, is a methionine, an amino acid that's called methionine. You will guys take it in the next semester in biochemistry, so don't worry about it now. Yeah, so roles of tRNA. We have mRNAs, tRNAs, and RNAs. I guess you guys took them. The messenger, what was it? Messenger RNAs. I'm not sure. Messenger, I guess. Then we have ribosomal RNAs. Okay, المهم, you guys know them أصلا. Okay, so messenger RNAs, I guess that transfers, so tRNAs and ribosomal RNAs. How is it shaped like? It's a handled drill. Why is it well suited for its dual function of binding to both an amino acid and an mRNA codon. The amino acid is bound to one end of the tRNA. At the other end is its anticodon. So it has 2 locations, the anticodon one and the amino acid one. That's why it's suited for its function. A 3-base sequence, the anticodon, which is a 3-base sequence which binds to the mRNA codon, calling for the amino acid carried by the particular tRNA. Anticodons form hydrogen bonds with complementary codons. tRNA is the link between the language of nucleic acid and the language of proteins. So if they ask you what's the link between the language of the nucleic acid and the language of proteins, which is the tRNA. If the mRNA codon is AUA, which specifies isoleucine, you don't have to know this. The tRNAs carrying isoleucine will have the anticodon UAU. So they are just saying that if we have, as an example, an mRNA codon that is AUCG, then the anticodon will be UAGC. That's it. Okay. Okay. The attachment process is controlled by an amino acid tRNA synthetase enzyme and is activated by ATP. So it needs energy, which is in the form of ATP. Once its amino acid is loaded, the tRNA, which is now called an amino acid tRNA. Why? Because it has an amino acid cargo. That's why it's called amino acid. So amino acid tRNA, it has with it an amino acid. So the tRNA diffuses to the ribosome where its amino acid is positioned. The ribosome is more than just a passive attachment site for mRNA and tRNA. Why? Because it holds all the process. تمام. The ribosome holds, oh my god, the ribosome holds the tRNA and the mRNA close together and coordinate the coupling of codons and anticodons. So we mentioned that no, we didn't. The ribosome has one binding site for mRNA and 3 binding sites for tRNA. Let me show you this. These are the 3 binding sites, which is A, P, and E site. Okay. So it has 1 binding site for mRNA and 3 binding sites for tRNA. The A site, which is an amino acid site for an incoming amino acid tRNA, and the P site peptidal site for the tRNA holding the growing polypeptide chain, and the E site exit site for an outgoing tRNA. It also has 3 phases, which is initiation, elongation, and termination, but it differs from transcription, that transcription happens in the nucleus, but translation happens in the cytosol or cytoplasm. We also mentioned that it requires energy in the form of ATP and a specific set of proteins that we're going to mention. Initiation, where a small ribosomal subunit binds to a special methionine carrying initiator tRNA and then to the new mRNA to be decoded. So أول شي بصير إنه the small ribosomal subunit it comes and binds with the mRNA carrying the initiator tRNA, then the new mRNA to be decoded with the initiator tRNA still in tow. The small ribosomal subunit scans along the mRNA until it encounters the start codon. So it runs along it until it finds the start codon, which is AUG, August. When the initiator tRNA is UAC anticodon, UAC is the anticodon for the AUG. Recognizes and binds to the start codon, a large ribosomal subunit unites with the small one, forming a functional ribosome. So when does a functional ribosome forms? When the small and large subunits are already together. تمام. أما قبل لما بتكون when we have only the small one with the mRNA and tRNA, it's not yet functional. We should have both subunits together. As this phase ends, mRNA is firmly positioned in the groove between the ribosomal subunits, and initiator tRNA is sitting in the P site, and the A site is free, ready for the next amino acid to deliver its cargo. So if they asked you, before we start, the initiator tRNA is positioned in which site, you should know that it's in the P site. Tamam, this is so important. I'm not sure if it came to us, but I think yes. So before it starts, the initiator tRNA is on the P site, which is the middle site, and the A site is free, ready for the amino acid to deliver another cargo. So going to the next phase, which is elongation, the ribosome moves along the mRNA in one direction, and one amino acid at a time is added to the growing polypeptide, as you guys can see here. شوي it comes, then it adds another amino acid, it adds more and more and more until it finishes. بصير في عنا a complete polypeptide. So codon recognition, the incoming amino acid tRNA binds to a complementary codon in the A site of a ribosome. خليني أفرجيكم إياها here. Okay. So the anticodon of an incoming tRNA binds to the A site. Okay? بعدين after it binds هاي الليوسين it binds then الليوسين بيروح على البوليبيبتايد chain و it's completed then it's transferred to the P site. After it's transferred to the P site, what was the tRNA that was in the P site is خلاص is released from the E site. Okay, so here they are saying that هون إنه it binds to the complementary codon in the A site و peptide bond formation and enzymatic component in the large ribosomal subunit catalyzes peptide bond formation between the amino acid of the tRNA in the P site and that of the A site هون here. okay, where it هون قبل ما تجي ال tRNA هون it's after it hydrolyzes the new bond تمام وخلاص بتقطع وبتجي the new tRNA again here. So yeah, between the amino acid of the tRNA in the P site and that of the A site. Now translocation, the ribosome translocates or moves shifting its position one codon along the mRNA as I showed you. This shift moves the tRNA in the A site to the P site. Why it's highlighted in a different way? Because it's important and it came to us in the final and we didn't expect it to come actually. So if they ask you the tRNA moves from where to where, does it move from A to E side, does it move from A to P side, does it move another options, how does it move? It moves from A to P side. The unloaded tRNA is transferred to the E site, which is the one that is gonna خلاص it's gonna be released. It's released from the E site, from which it's released and ready to be recharged with another amino acid. This process continues. The tRNA in the peptidal site transfers its polypeptide cargo to the incoming amino acid tRNA, and then the P site to E site and A site to P site movement of the tRNAs occurs. The initial portion of the mRNA passes through the ribosome and may attach successively to several other ribosomes, all reading the same message. This multiple ribosome mRNA complex, a polyribosome, efficiently produces multiple copies of the same protein. So this is why we have exons and introns, because here in an mRNA I meant that إنه هاي ال mRNA كاملة فبتجي ريبوسومي لهون and then it moves from the promoter to the initiator. بتجي ريبوسومي لهون promoter to initiator. So we have multiple ribosomes on one mRNA and that produces multiple copies of the same protein. So for termination, a stop codon, we mentioned them, which are UAA, UGA, and UAG, enters the A site. The stop codon is the period at the end of the mRNA sentence, which it tells you that خلاص, let's end this, we finished. It tells the ribosome that translation of the mRNA is finished. As a result, water instead of an amino acid is added to the polypeptide chain. This happens why? Because it hydrolyzes the bond between the polypeptide and the tRNA in the P site. خلينا نفرجيكم الصورة itself هون. اوكي, وصلنا للستوب كودون. بعد ما نوصل للستوب كودون تجي الووتر خلاص, هذا هو release factor. It binds here to the stop codon, it hydrolyzes this bond وخلاص ال polypeptide chain detaches و it's ready خلاص. okay, نرجع. Let's go back to this. Water instead of an amino acid is added. It hydrolyzes the bond between the polypeptide and the tRNA in the P site. The completed polypeptide chain is then released from the ribosome, and the ribosome separates into two subunits. The released protein may undergo processing before it folds into its complex 3D structure. When the message of the mRNA that directed the formation of protein is no longer needed, the mRNA is degraded. So, hon, they are telling you that after we hydrolyze the bond, both subunits of the ribosome detaches. mRNA, if we don't need it, then it's degraded. That's it. And the protein may be folded to a 3D structure. To summarize the process here بالfigures, translation begins with the mRNA binds to the small ribosomal subunit. The amino acid is brought to the ribosome by a tRNA, then the initiator tRNA binds to the start codon, then the large ribosomal subunit binds to form the translation complex, and initiation is complete. Now for elongation, the tRNA molecule binds to the A site هون. tRNA comes in, it binds to the A site. A peptide forms between its amino acid and attached to the tRNA at the B site هون. Then it slides and the nucleotide tRNA is released in the E site. So as I just mentioned, tRNA comes to here, then the bond forms between this and this, then if it wanna leaves, it comes to the E and then it's released. شو كمان when a stop codon is reached and it's binded to the A site, elongation خلاص, it ends and the polypeptide is released in the P site. So نحن شو حكينا, stop codon comes and binds to the A site, but actually the polypeptide chain is released from where? From the P side. So if they ask you, the stop codon comes and binds where? It binds to the A side. The polypeptide chain, it's released from where? It's released from the P side. The tRNA that is خلاص, we don't need it, is released from where? From the E side. We should all know this, okay? A site, what happens? Stop codon comes, ends the process, binds to it, خلاص, closes the place. No more tRNAs come. And the polypeptide is released from the P site. The tRNA is released from the E site. هذا أهم شيء. Okay, so let's go to this paragraph. I don't remember that it came, but let's go through it. Ribosomes attach to and detach from the rough endoplasmic reticulum when a short leader peptide called the ER signal sequence is present in a protein being synthesized. So when the protein is being synthesized, there is an ER signal sequence, which is the associated ribosome attaches to the membrane of the rough ER. It's guided to appropriate receptor sites on the ER membrane by a signal recognition particle, which is a ligand, a protein that cycles between the ER and the cytosol. هون عم بفرجونا ال ER signal sequence. So the SRP directs the mRNA ribosomal complex to the rough ER. There the SRP binds to a receptor site. هاي receptor site وهي ال SRP itself وهي ال sequence. They bind to the receptor site. Then once it's attached to the endoplasmic reticulum, the SRP is released. خلاص, it's released, and the growing polypeptide snakes through the ER membrane pores into the cisterns. These are the cisterns. كل هاي cisterns, تمام? You all know this, I guess. An enzyme clips off the signal sequence as protein synthesis continues. Sugar groups may be added to the protein. هاي ال polypeptide chain, هاي ال sugar groups. The complete protein is released from the ribosome and folds into its 3D conformation. هاي is 3D conformation. The protein enclosed within a vesicle. هاي ال vesicle. The transport vesicle makes their way into the Golgi apparatus. هاي ال vesicle, it goes to the Golgi where further processing of the protein occurs. And that's it for this paragraph. And we also have this small one, which is talking about the rules of DNA. We mentioned here that what differs is that the mRNA has uracil, but DNA has thiamine. Okay, for the roles of DNA, that it also produces other active RNA species that are not translated into proteins, including micro and small interfering RNAs. MicroRNAs, from its name, micro, small RNAs that interfere with the suppress mRNAs made by certain exons. What does it do? It silences them. So the keyword is silencing them. What does it do to them? Micro RNAs silence them. إذا إجاكم سؤال وشفتوا silencing them, خلاص micro RNAs directly. Small interfering RNAs are like micro RNAs but originate outside the cell. Our cells can use an infecting virus RNA to create small interfering RNAs that then interfere with viral replication. So what do small interfering RNAs that are originating outside the cell and they have to do with the viral replication. اوكي خلينا نحل هالسؤالين. I'm just checking if there's other notes that I didn't mention. So here the ligand that we talked about, which is the SRP1, the signal recognition particle, without it no protein will form, so it should be there for the protein to be formed. Okay, so how do the AP and E ribosomal sites differ functionally during protein synthesis? A is the entering site, P is the growing site, and E is the exit site. تمام. What's the role of DNA in transcription? It's the template for protein synthesis. Oh, that's it. If you have guys any question, you can message me after I send the video, and I hope it was helpful. I really hope it's helpful, and if I miss anything or if something wasn't that clear, you can ask also. And good luck on your cell quiz. Inshallah it will be easy. But thank you guys for listening.